I'm really happy to introduce Heather. We are at the same institution. So Heather Pua is an assistant professor in the Department of Pathology, Microbiology, and Immunology at Vanderbilt University Medical Center. We're here in Nashville, Tennessee in the United States. So Heather is a physician scientist, and her research focuses on the role of small non-coding RNAs and extracellular vesicles in tissue inflammation. And she has a particular focus on the lung during allergic responses such as asthma. And she also, in addition to her research, does clinical work as a molecular pathologist. And so she has a long-term interest in how novel molecular assays can be translated to help diagnose and treat patients with chronic inflammatory diseases. So I'm really excited to be able to hear what Heather has to tell us today.
All right, well, it's really my great pleasure to be invited to speak at the WebEVTalk. And today I'm going to tell you a story about, sorry, it shouldn't just be human, it should be human and mouse extracellular vesicles, origin and heterogeneity and inflammation. And as Alissa said, I'm here at Vanderbilt University Medical Center in Nashville, Tennessee. It's a beautiful time of year. It's just starting to be fall colors, and it's a great time. So the work that I'm going to tell you about really is work that I began during my postdoctoral fellowship at UCSF with the help of a really talented technician, Hannah Happ, who is now a graduate student up in Chicago. And it's continued here in my lab at Vanderbilt, both through the combined efforts of Carly Gray, who was a technician in my lab, and now sort of continued by Katie Bunn, a really talented graduate student. As Alissa mentioned, one of the interests of my lab is asthma. And asthma is a chronic inflammatory disease. It can actually affect all age groups. It often presents in children, but some forms of asthma will present in adulthood. And although some people will have really sort of get over their childhood asthma, many have persistent disease. Interestingly, if you look across the world, asthma is the most common chronic lung disease worldwide, and the prevalence of asthma is really quite amazing. So probably everybody on this call knows somebody with asthma, because the prevalence of disease, depending on the country, ranges from about 2 to up to 15%. So in the US here, about 7 to 8% of the population has asthma. Clinically, asthma is characterized by reversible airflow obstruction and bronchial hyperresponsiveness, but sort of at a cellular and molecular level, asthma is really characterized by an allergic inflammatory response in the lung. And this diagram is sort of a simplification of the type of allergic inflammation that we see in asthma. And when an individual encounters allergens in the environment, this can include things like house dust mites, cockroach antigens, pollen, fungal spores, hair or dander from pets. There's activation at the airway epithelium or the respiratory epithelium, which sends alerting signals to the immune system and really activates a really robust inflammatory response. And this allergic inflammatory response is called a type 2 immune response and is really characterized by the activation of T cells, by dendritic cells, and subsequent production of IgE from B cells. There's recruitment and activation of eosinophils, and eosinophils are one of the hallmarks of allergic inflammation, as well as activation of innate lymphoid populations. In addition, these IgEs can then charge mast cells to respond to these allergens very rapidly on re-encounter. So there's a very complex and dynamic process that occurs along respiratory epithelium during this allergic response. And once this response occurs, there really are very dramatic changes in the lung that occur. On the left-hand side, you can see a normal airway. So you have nice alveolar sacs And sort of the outlying tissue here, and in the center, you have a bronchial epithelium with a bit of pink smooth muscle and a little bit of matrix around it. On the right-hand side, you have a patient who's had long-standing chronic asthma. You can see that there's a real thickening of this interstitial space with a lot of increase in fibroblasts and collagen. The muscle has also thickened around this airway, and you can see a lot of metaplasia of these epithelial cells, which now become really robust mucus-producing cells. And we understand that many or most of these changes are really driven by this chronic inflammation that's occurring in the tissue. So when I sort of started working on this, I had been working on microRNAs and really their ability to regulate intrinsic cell programs in immune cells. And we started to get really fascinated by the literature coming out showing that there were a bunch of these microRNAs in the extracellular space. So we started asking a really simple question, which is whether inflammatory extracellular microRNAs increase with allergic lung inflammation. And the biofluid that we were interested in sampling was this airway lining fluid here depicted in blue, which really lines the bronchial all the way through the alveolar space. And in order to pick up or detect this airway lining fluid, we perform a process called bronchoalveolar lavage, where we shoot some PBS down into the lung. It distributes throughout the airways and the alveolar spaces, and then we can wash it back up. So unlike some other biofluids that might be collected, like cerebrospinal fluid or plasma or serum, this is a diluted biospecimen because we can't directly sample this airway lining fluid. I mentioned that we were interested in microRNAs, and probably everybody in the audience knows what a microRNA is, but just to make sure we're all on the same page, microRNAs are short, non-coding RNAs. They're typically 21 or 22 nucleotides in length, and this is just about as long as a common, primer you might design in lab to do some kind of PCR assay. They're transcribed by the same RNA polymerase that transcribes mRNAs, and they undergo a set of sequential processing steps from transcription in the nucleus until they reach a mature form in the cytoplasm. And here they really will complex with argonaute proteins as well as other accessory proteins that will help to target them to the 3' UTR of mRNA transcripts. And after binding to this 3' UTR, they can actually inhibit gene expression. So they're classified as post-transcriptional inhibitors of gene expression. And typically we think that they function by destabilizing these mRNAs, although there is some evidence to indicate that they can also inhibit translation of mRNAs into proteins. So we began our experiment by collecting both biofluids and cells from mice with a model of allergic airway inflammation. So we had two control groups, one which we called our CTR control mouse, which really had no manipulation done to it. A second group of mice that were immunized intraperitoneally to induce a systemic inflammatory response, but had no challenge in their lung to recruit this allergic type inflammation into the lung tissue. And finally, our mice with allergic airway inflammation, who were both immunized and then challenged in the lung. We immunized with a model antigen called ovalbumin, along with an adjuvant alum that's known to induce sort of a robust type 2 inflammatory response. And then the mice are challenged in the lung for 3 days to recruit these immune cells into the lung. We decided we would go ahead and take two biofluid specimens, so this bronchoalveolar lavage fluid, as well as serum from our mice. And we also took two cellular materials that we thought would be likely to be sources of microRNAs in this fluid, which includes the cell pellet that comes down from the BAL fluid wash. This is typically very rich in macrophages at steady state and then contains a mixed inflammatory infiltrate in the allergic lung. In addition, we did epithelial brushings in our mice to pull off this bronchial epithelium that is really kind of the source and interface of where the allergic reaction occurs. So we isolated RNAs from our sample, and this is a bioanalyzer tracing of some of those RNA samples, and very consistent with what was known and has been published in the literature. We could see in both of our fluid samples, our BAL fluid and our serum, we have a sort of dominance of small RNA species, while as in our epithelial cells and our pellets from the BAL fluid, we can see nice ribosomal RNA subunits consistent with cellular RNA sources. We went ahead and did small RNA sequencing. We generated our own libraries to do this and then sequenced them. And here on the right-hand side, I'm showing you the percentage of mapped reads, in red being microRNAs, which is the predominant species that we recovered, as well as a few other smaller species subtypes. The first thing we noticed was that microRNAs are pretty abundant in BAL fluid. So here's a comparison in panel A of the femtomoles of microRNA compared to the total micrograms of input RNA. We did a normalization using a series of calibrators in our sequencing that are not expressed in mammalian systems. And we could really see that there was, a fairly high content in our BAL fluid, somewhat similar to our cells, a little bit lower than our serum, but remember that this is a dilute biofluid. In addition, we looked at what we anticipated to be the microRNA copy number per microliter of fluid and saw in the order of 10 to the 6th to 10 to the 7th copies per microliter of BAL fluid. Again, slightly lower than in serum, but we think that BAL fluid is probably diluted at least 10-fold from the original airway lining fluid. Next, we wanted to actually see what is the sort of relative composition and association between these different RNA groups. And so we performed a principal component analysis. And I think, two things are sort of initially striking from this data. So clearly these pink group of dots, which were our serum, are pretty far divided from all of the samples coming from the lung. And this just confirms, that, there is not a single signature for all biofluid microRNAs, and that it probably has a lot to do with the cell types that are encountered with this biofluid and that are providing its sources. So serum and BAL fluid look quite different, but BAL fluid certainly had some similarities both with the pellet from the BAL fluid, which contain immune cells, as well as with the epithelial cells. And on the right-hand side, this is just an alternative representation of our data showing the Pearson correlation coefficients between the BAL fluid and epithelial cells and pellets. And you can see that the tightest correlation was between BAL and epithelial cells. Not terribly surprising because epithelial cells will be the most abundant cell type in contact with this biofluid. Next, we asked a question of whether the total microRNA content actually changed with the induction of allergic airway inflammation. So again, our CTR and our immunized are our two control groups, and our AA is our allergic airway group. And really in the BAL fluid, we didn't see any changes in the total quantity or copy number of microRNAs detected before or after inflammation. Well, interestingly, we did see a trend towards increase, I would say, in the serum of microRNA copy numbers with either systemic or systemic plus the localized inflammatory response. But what we could definitely see is that there were a subset of microRNAs that are increased in the BAL fluid during allergic inflammation. So in this plot, we've plotted base mean expression of the microRNAs here on the x-axis against the log 2-fold change, comparing our BAL fluid from our allergic mice to both of our control groups. And most of the microRNAs do not substantially decrease, suggesting that there's not a total shift in the microRNA content in the extracellular fluids. But we do see select increase of microRNAs. And what we began to notice as we look at the microRNAs that increase in the allergic lung, microRNAs such as miR223, miR142, AA3P, and 5P, miR-150. These are all microRNAs that we know are both abundantly expressed in immune cells and have really important roles in regulating immune cell function. So this started to give us a sense of a picture that perhaps part of the inflammatory program was that during tissue inflammation, that immune cells that were recruited into that space were actually spitting out microRNAs. The other reason that we got really very excited about this data is because of some published human data. So at the time that we did this study, there were 2 studies that actually suggested that in humans as well, extracellular microRNAs could be changed in respiratory fluids of patients with asthma. In one study, they looked at the differential expression of extracellular microRNAs from induced sputum of severe asthmatic patients. And in another study, they looked at differential expression of extracellular microRNAs in 100,000 GBAL pellets in patients with mild asthma. And in this top study, where they were looking at induced sputum of patients with really severe asthma, what was really striking is that at least 2 of the 3 microRNAs that we identified as upregulated in our mouse model are also seen as increased in patients with severe asthma. And so that really suggests that perhaps our mouse model is recapitulating some of what's occurring in the human pathology. So, you know, we have had these extracellular microRNAs, and, you know, at first face, it seems a little bit surprising that there are microRNAs in the extracellular space. And so we really wanted to understand how these microRNAs were packaged. And so the first question we asked was a fairly simple one, which is whether the extracellular microRNAs in the BAL fluid are stable or they degrade. We know that there are RNases actually present in all biofluids and many surfaces that sort of serve as part of the ancient viral defense. So we did an experiment where we actually extracted RNAs from BAL fluid, and that's the black circles here that are the RNA pellet. And then we took just freshly isolated BAL fluid from our mice, and we added increasing amounts of RNase to these 2 samples. And what you can see is for our RNA pellet, we get a nice dose response as we have increasing amounts of RNase. We basically are capable of degrading our RNA, but it's really quite striking that even when you add very large amounts of RNase to BAL fluid, you don't really see much of a decrease in classic microRNAs that we found expressed there, such as miR-21 or miR-24 or miR-223, which we said was upregulated. And here's miR-34, which is expressed by epithelial cells. So this certainly suggested that extracellular microRNAs in the lung were being packaged in some way to protect them from degradation. And so, of course, the first and logical thing to think about was whether they were being packaged in extracellular vesicles. So we went looking for extracellular vesicles in the BAL fluid. And by performing nanoparticle tracking, we could detect about 10 to the 8th particles per microliter in BAL fluid. Again, this was about 10-fold less than we see in serum, but BAL fluid is a dilute biospecimen. The size of the particles that we detected was typically 200 nanometers and below, and was slightly larger than the particles that we saw in serum. So we performed serial ultracentrifugation, prepared 100,000 G pellets, and performed electron microscopy. And indeed, in our BAL fluid samples, we could see particles that had classic EV morphology. So this is an approximately 100 nanometer vesicle. It has this sort of donut-shaped morphology. However, from the BAL fluid, there are also many liposome-like particles, and that's depicted below. They had a little bit more variable size, but certainly could be around 100 nanometers in size. And by electron microscopy, they really looked like lipid monolayer. They comprised about 90% of the particles that we could detect, whereas about 10% looked to be of this classic EV morphology. And what we think is that these liposome-like particles are probably surfactants. So in the lung, at the surface of the air-liquid interface, there's actually a monolayer of lipid with studded proteins. And this has probably lots of important roles, but one of the most important early roles is that this decreases surface tension and actually allows inflation of the lung. So what about protein markers? Could we detect protein markers of EVs in our fractionated BAL fluid from mice? So on the right-hand side, I'm just showing you a Ponceau stain to give you a sense of overall protein loading from our cell pellet from 2 BAL fluid samples prepared at 100,000 G and 2 serum samples similarly prepared. And what you could see is that indeed we can certainly detect many of our classic EV markers in these cell pellets from the BAL fluid, including ALIX, TSG101, CD63, syntonin, and ARF6. So what about packaging of these microRNAs into this EV fraction? So the way that we approached this is that we performed serial ultracentrifugation followed by flotation into a sucrose gradient, and we floated up and collected a fraction that floated at 40% sucrose. And I think that this was really quite interesting and informative. So in clear, you can see our immunized mice, and the shaded bar that you see is our allergic mice. You can see that microRNAs can be detected in this floated fraction. And some microRNAs are unchanged, such as miR21 and miR34C, but the microRNAs that we detected as upregulated in our RNA sequencing data of total biofluid are also increased in this EV-enriched fraction, and that included miR223 and miR142A, where we didn't see differences maybe in fragments of ribosomal RNAs or U7 snRNA. So, we were kind of interested in we couldn't really detect a change in the amount of microRNA in the allergic lung. So we wondered if actually the total number of particles might increase in the allergic lung. And so we again performed nanoparticle tracking to look at mice that had been immunized but not challenged in the lung versus mice with allergic airway inflammation. We really saw no change in total particle number. We also couldn't detect any changes in the size of particles. So, that really suggested that if there are changes in EV composition, then it is likely that it is a shift in the type of EV present or a subset of EVs that we're not sensitive enough to detect by these bulk measures. We've been really lucky since coming to Vanderbilt. We have a lot of great collaboration between basic science, translational science, and clinical science. And we've been able to actually get some samples from a collaborator in pulmonology here, Ryszard Dworsky. He has a group of patients who have allergen-induced asthma, and he will bring them in for a visit and do a baseline BAL fluid in these patients, followed by a segmental allergen challenge where he actually instills allergen into a subset of their lung. We'll then bring them back for a second visit post-allergen challenge and retake a BAL wash. He provided us some samples from these 2 BAL specimens for a group of patients. So just as in our mouse, when we tried to look at total particle numbers, either in our unfractionated BAL fluid or our serially fractionated samples, either at 1200 G, 10,000 G, or 100,000 G, we really couldn't see any difference in baseline samples from individuals to allergen challenged samples from individuals. We were able to detect classic EV markers in this 100,000 G pellets that were really not present in the 10,000 G pellet, including ALIX, TSG101, and syntonin in the human samples. But when we quantified sort of the relative intensity of these protein markers, again, as a potential surrogate for how much EV material might be there, we didn't really see any consistent changes, although you can appreciate there's quite a bit of variability in the allergen challenge group. with one patient in particular having very high protein levels. So clearly looking at this bulk measure of how many EVs were there was not going to really answer a question about what populations of EVs might change. And because we saw that microRNAs from inflammatory cells were increased, we really thought that there should be immune-derived EVs present in this space during the tissue inflammation. So we wanted to know whether we can trace the origins of either the extracellular RNAs or the EVs in vivo during allergic inflammation. Tracing RNAs in vivo is very challenging, but we thought that maybe we would have a chance to actually trace the origin of EVs in vivo. And the way that we approached this is that we basically took a commercially available mouse to try to develop a cell type specific membrane reporter to trace the origin of EVs by flow cytometry. And in this mouse, they express constitutively a membrane bound tomato that is surrounded by 2 LOXP sites. And so in the presence of a tissue specific Cre recombinase, you can actually get excision of the membrane bound tomato and expression of a membrane-bound GFP. And what we thought that this might allow us to do is to label cells either green or red, depending upon which tissue-specific Cre we chose. And if we were able to visualize extracellular vesicles from those 2 cell populations, we may be able to see green and red subpopulations of EVs that would allow us to back trace the EV to its origin cell. And we were going to approach this by doing EV flow cytometry. So this GFP is actually an eGFP that was fused to a small sequence of Mark's protein, which becomes both muracylated and pomidylated at a canonical sequence in that protein. And when we take BAL fluid from our mice and perform 10,000 and 100,000 G ultracentrifugation steps, we can see that we can detect GFP only in the presence of Cre recombinase in both 10,000 and 100,000 G fractions, suggesting that yes, indeed, that we're getting some segregation of this membrane-bound GFP into vesicles. So the initial work on the flow cytometer for this project was actually done back at UCSF. We used a Cytoflex instrument. And so the first few plots I'll show you there, but since coming to Vanderbilt, we've switched instruments due to availability. And so here you can see on the Cytoflex where we just ran PBS, there's definitely a background here that is positive on side scatter, but does not fluoresce. And we can certainly detect beads down to 100 nanometers. Of course, their refractive index is not the same as an EV. But at least it was reassuring to see that. And so we looked at the BAL fluid of wild type MTMG mice and MTMG mice that had been crossed to an actin Cre promoter, which should be ubiquitously expressed. So pretty much all of the tomato signals should be changed for GFP. And as we would expect in our wild type situation, we don't see basically any fluorescent positive particles. In our MTMG mice, we only see membrane-bound tomato positive particles in the red channel. And in our MTMG actin Cre mice, we only see green positive EVs in our MGFP channel. So it seemed that the system was working. So we went ahead and we crossed our MTMG mice onto 2 Cre lines to begin with. And the 2 Cre lines we chose were an NKX 2.1 Cre, which is a transcription factor that's actually expressed early in lung development and should really label all lung epithelial cells. And that's what's depicted here. You can see labeling of both the bronchial epithelium, alveolar epithelium, and these sort of plump cells are type 2 alveolar cells, which are secretory alveolar epithelial cells. And then we also crossed our MTMG mice to the VAV Cre mice, and VAV is expressed early in hematopoietic cell development, so it should really label kind of indiscriminately all immune cells. And we can see a nice immune cell population here infiltrating next to the bronchiole, probably coming out of this vessel here, as well as patrolling alveolar macrophages. So when we looked at our 2 mice lines at steady state, we could see really a very robust population of GFP positive EVs in our MTMG NKX 2.1 cream mice, suggesting that many of the particles that we could detect were coming from the epithelium, very analogous to what we saw with our microRNA data. When we looked at our MTMG VAV-CRE, we could definitely also see a population of GFP positive particles, though obviously they were much less in number. And when we did quantification, we could see that about 80% of the fluorescent positive EVs that we detected seemed to be coming from epithelial cells, and somewhere between 10 and 15% at steady state were coming from our immune cell population. So that in and of itself was really interesting, but what was most exciting was that when we looked in our inflamed mice, we saw a trend towards an increase in our epithelial EVs here in the allergic airway, MTMG, NKX, CRE positive mouse. But we saw big increases in the number of EVs detected of immune cell origin, and on average about a 3-fold increase. So again, it's sort of very consistent with this idea that during tissue inflammation, one of the properties or functions of immune cells is that they begin to secrete these immune EVs, and we believe that they're in part packaging RNAs. So I'm really excited because here at Vanderbilt, we've kind of tried to take this system to the next level. This is work by Katie Bunn in my lab, the graduate student. So she's gone ahead and crossed this MTMG mouse onto an LCK CRE mouse. So this CRE will only label T cells in particular. And we know that T cells are really important drivers of this chronic allergic inflammation. So here's her kind of nice immunofluorescent picture. This big structure on the left is the bronchiole. Here you can see a vessel, and then this sort of space around the bronchovascular bundle. And you can see lots of nice little T cells here infiltrating in this space. The plots look a little bit different here because we've switched over instruments, and we're now triggering on fluorescence rather than scatter. And so some of that background is gone. But what you can see is in the immunized mice, when we look at the LCK CRE MTMG versus the MTMG mouse, really in neither case do we see a population of GFP positive vesicles. However, in the allergic lung, we can see that about 3% of all fluorescent positive vesicles are now carrying the GFP tag, suggesting that yes, indeed, that T cells come in and they secrete extracellular vesicles. When Katie quantified the T cell EVs in her airway lining fluid, she could detect about, between 1,500 and 2,000 T cell EVs per microliter of BAL fluid. And when we compared the number of GFP positive particles recovered to the number of T cells that we actually recovered from the tissue, which we did simultaneously, she could detect about 30 T cell EVs for every T cell that we detected in the tissue and in the BAL fluid. Okay. So I think that this, this has been a great and quite powerful system for us to begin to really pull apart and detect different subpopulations of EVs. So the question is, how do you actually then apply that or kind of use that knowledge to begin to ask questions in human populations? And can we actually find human immune EV subpopulations? Obviously, there are no transgenic people who have fluorescently labeled EVs, so we have to take a different approach. And the approach that we've kind of settled on for now is using a bead-based capture kit and using bead-based flow cytometry for assessing EVs from human biofluid. So this is a schematic that's really adapted from Miltenyi, who produces a MaxPlex kit. This is a kit where there are a series of beads that are hard dyed in the FITC and PE channel with varying intensities. And this allows you to segregate different bead populations. And then each population is bound to one of 37 different capture antibodies. This was an assay that was really, I think, initially designed by the company for detection in plasma. So it has a lot of immune cell markers, which we're of course very interested in. So you basically will bind EVs onto your beads and then use a secondary antibody to one of the many EV tetraspanin markers. In our case, we have used CD63 and CD9. And that basically allows you to detect EVs bound on the surface of these capture beads. So this is sort of a fancier modified sandwich ELISA. So when we were getting into this system, the first thing that we did is we looked at some purified Jurkat EVs, and Jurkat cells are a human cell line that has some features similar to a CD4 T cell, but it is a leukemia cell line, so it doesn't behave in the same way that a primary T cell would. On the left-hand side, you can see our gating of forward and side scatter for the beads. We then can array the bead populations by gating FITC versus PE, and then looking at APC to detect how many EVs are present on surface of beads. And here you can see either with capture beads or media alone, we really don't detect any EV populations. But if we purify Jurkat EVs, you can now see nice APC population here appearing. And then you can go back and you can look at each of these individual bead populations and determine what types of EVs you can see. So this was really the results of those first experiments displayed as a heat map. So on the top, we have two negative controls, which is basically the isotype controls for bead capture. So first, if you look at EV markers, you can see that you really get no capture with just beads or media alone. And that Jurkat, these Jurkat T cell lines really express very high levels of CD81 and relatively high levels of CD9, much lower levels of CD63 in their EV populations. In addition, they have markers on their EV that are what we would call classic T cell markers, such as CD45, CD4, and CD3. And they're negative for markers we would expect that they shouldn't have. So something like CD8, these are supposed to be a CD4 positive T cell lineage, and CD19, which is a B cell marker. Then it's quite interesting because they also have these additional positive markers, which we don't really understand what a function would be, but they express additional markers such as CD29, CD24, 49E, 31, 146, and a little bit of MHC class 1. So you can sort of get a type of sort of profile or fingerprint of the EVs produced, at least by this one cell line. So we wanted to be able to apply this to some patient samples. We've tried a couple of different iterations. I'm going to show you the one that we've had success with the most so far, but this is super early data, so still a lot of work to do. And the group of patients that we've been looking at are a very small, not very small, but a smaller subset of asthmatic patients who have a particular disease entity called aspirin exacerbated respiratory disease, or AERD. And these are patients with underlying asthma and chronic rhinosinusitis that have symptoms that are very exacerbated by NSAID administration, such as aspirin or, in particular, aspirin. They have recurrent nasal polyps, and they really have both upper inflammatory symptoms and lower inflammatory symptoms. And what's interesting about this patient group is that they tend to present in adulthood, unlike maybe the most common forms of asthma, which present in the pediatric population. And they more commonly have a very severe and treatment refractory disease. And it's really known that, at least in part, the underlying pathology involves a dysregulation of arachidonic acid metabolism, which is exacerbated when cyclooxygenase 1 is inhibited by an NSAID. These nasal polyps that they get contain mast cells and eosinophils, neutrophils, and leukocytes, and so all of these kind of classic cells of the type 2 inflammatory response. So this was work that has been done in collaboration with another pulmonologist here at Vanderbilt, Catherine Cahill, And she's actually the one who approached me about looking at the sample type. And in honesty, I didn't think it would work, but it works great. So she actually puts nasal sponges into the nose of her patients, and these sponges will absorb the fluid as they're having some type of reaction. What's really nice about this patient population is that you can take a baseline measurement, you can challenge them, and then you can kind of start a clock when the patient begins to report symptoms and take serial samples as they go to the peak of their response, which usually occurs between 60 and 120 minutes. And then usually the response starts to go down around 180 minutes. So the first question is whether we could detect particles from nasal sponge specimens. And so again, we did nanoparticle tracking, looking just at a sponge alone versus a sample. So a sponge that had been placed in someone's nose, and then the fluid had been spun out of the sponge. And indeed, like, we could certainly detect particles around 10 to the 10th per mil in these nasal sponge specimens. The particle size here looks a bit bigger than we can see sometimes in our BAL fluid specimens or in our plasma or serum specimens. And this is a super complicated slide, so we'll walk through it, but I think it has some really interesting and exciting data. So again, on the top, I've presented the negative controls. So these are the two basically isotype controls for the bead capture. On the left-hand side, there's another set of controls, which is our bead only and our sponge. And then a plasma sample kind of serves as a positive control to make sure that we could detect some markers in the run. And what you can see is that we can trace the basically the response of the patient from time 0 to time 180 over the course of this response. And we really get a couple of very unique patterns of response among what looks like different EV subpopulations. So the first is that we seem to have this early peak at time point 0. So basically when a patient first starts to report symptoms, we see about a 2-fold increase in EVs that carry this marker CD133. Then as patients are really reaching the peak of their response, so in this patient, the peak was at 160, but patients will sort of vary between 60 minutes and 120 minutes. You can see now that we have a population of CD9, CD81 positive EVs that are peaking, they're going up and then coming down a little bit after this 60-minute time point. That also carries some other markers, including CD142, CD326, CD44, as well as some additional markers. And then finally, when we look at CD63, which is often seen as a kind of classic tetraspanin surface marker for EVs, we can see that the pattern is totally different than what we see for CD9 and CD81. There certainly is an increase in the number of particles in patients as they have a reaction, but it doesn't peak at the 60-minute time point. And in fact, if anything, it looks like it slowly trends up even through 180 minutes. Now, you know, we're immunologists, so we would have loved it had we seen our T-cell markers, for example, or some other kind of classic immune cell marker. But I have to say that really what we think we see when we try to look at some of these markers using the Human Protein Atlas, which has taken autopsy samples of all types of tissues across the bottom and done across the whole body and done staining, is that many of these markers, including CD133, CD326, which is also known as EpCAM, and CD44 are really abundantly expressed in the nasal epithelium. So we actually think that this big burst of EVs that we can detect in this nasal fluid is likely coming from epithelial cells, and that this may actually be a response to immune cell signaling that we know is occurring at this time inside the tissue. So in conclusion, hopefully I've shown you today that extracellular microRNAs change during allergic lung inflammation with select increases in microRNAs, including miR223 and miR142A. We've shown that EVs or EV-enriched fractions can carry microRNAs in the lung. We can trace the origins of EVs using a fluorescent membrane reporter system coupled with EV flow cytometry. And most of the EVs that we can detect, at least, are of epithelial origin, but there's really a select increase in immune EVs in the allergic lung. And we're beginning to think that we see dynamic changes in human EVs that occur in response to activation of local allergic tissue inflammation. So I'm never entirely sure what to put about in future directions for EV talks, because I think we have nothing but questions. There's so many interesting potential future avenues to study with EVs and extracellular RNAs. Everything from very basic questions about, continuing to understand cellular sources and the regulation of their export, trying to understand which RNA containing vesicle subsets there are, or whether many vesicles contain RNAs, what their function is in the immune system, who they're targeting, and of course, what therapeutic implications this might have. My last slide is my acknowledgements. I would like to thank our funding sources and all the members of my lab. I think I mentioned along the way the folks who are doing the work. Again, this work was begun in my postdoc lab at UCSF, and we've had a lot of great collaborators, especially here at Vanderbilt, our clinical collaborators, again, who have given us these really invaluable clinical specimens, as well as folks in the EV community, especially Alissa, lets us do all this particle counting, and other folks here at Vanderbilt. So I'm happy and welcome any questions.
Great, okay. So I think I'll just start going from the chat box. So Phil, you had a number of questions in there. Would you like to ask some of them?
Well, I'm a great fan of this area, and this is very, very careful, wonderful, technically high-class work. And there's a million directions to go into. I think my first question was it was missing ribosomal fragments, which is a surprising part of things. But I think later on you showed that there's a lot of those, and we don't know what they do, but there's a lot of those. I think the other question asked was about time course, which you got to with the Samter type samples. And if you looked at the tetraspanins, you have two different populations of EVs, and there's a 60-minute peak and a 180-minute peak, and if you do that in the allergic sense, there's a lot. And then lastly, a lot of immune inflammation doesn't have many T cells, and so don't be disappointed about that. They are great recruiters. They're great recruiters. So that makes a sense too.
Yeah.
So this is, and the last is, you're in a wonderful environment for prostaglandins and leukotrienes. And I think there are very important stories that were begun by Suzanne Gabrielson in Sweden in this area that you really could get at with fantastic collaborations that you have at Vanderbilt. Thank you very much for this wonderful data.
Yeah, sure. I'll just make a few kind of fun kind of remarks about all this. Sure. Is, I think for the sequencing data, because, we require 5' phosphates is a very particular type of sequencing. I think really we were very biased towards capturing microRNAs. It's not to suggest at all that those are the only small RNAs or RNA fragments present in the allergic lung. I think there's many more, and actually probably ones that are more abundant than microRNAs either. So I think that's a very exciting future direction for this work as well. These populations of EVs are really fascinating, and it certainly makes me want to go back, and we've been talking about this, Katie and I have been talking about this, is also going back to our mouse model and thinking a little bit more about the subpopulations of EVs present and the kinetics of that response or reaction. And T cells is only because I started life as a T cell biologist, so I just have a special place in my heart for T cells.
Me too, but it doesn't take many. Also, yeah. You didn't have microRNA 155, which is a Treg marker, but lots of, in lots of immune inflammation.
It is, and I sort of wonder, the immune inflammation that we used in the asthmatic model is really a very acute, it's really an acute type 2 inflammatory response. It's not very chronic. And so I wonder a little bit if we went back to a more chronic model, something like a house dust mite model, where we really are inducing the inflammation in the lung, and then kind of chronically re-stimulating it actually might change the profile, things like 155.
I do them all. Time course is fundamental to learning things, and you've got it already. Thank you.
So just to follow up sort of on your allergic models, Heather, what, it seemed like in the nasal model, you were getting epithelial EVs, and maybe in the bronchial alveolar lavage, you were getting more immune cell EVs. Could you sort of comment about, you know, the different models there?
Yeah, well, I think that there's one very fundamental difference between that form of pathologic human inflammation versus the mouse model. So in the mouse model system, you really get a transudate. So you get both a transudate and an exudate in the mouse model. So you basically get immune cells that emerge from the tissue. Actually, they crawl out between the epithelial cells. They really get into that fluid because they're really trying to encounter pathogens before they even really gain access to the lung. But in the nasal situation, that doesn't seem to be the case. So in this very specialized kind of sort of asthma and allergic inflammation in the nose, it really is mostly thought to not be very cellular in the nasal cavity itself. And I sort of wonder if that's potentially part of the difference, or maybe it's also just a difference in the way the bronchial epithelium acts versus a nasal respiratory epithelium acts. I think any or all of those might be involved.
They're very different situations.
I think, Dolores, you had a couple of questions, including one about the cells in the nasal sponge.
Yeah. So my first question was, so you showed the RNases experiment. So you compared the isolated extracted RNA with the biofluid. Did you take in account the dilution factor in the biofluid?
Yes. So basically what we did is we tend to extract from 500 microliters of BAL fluid. We would extract that down all the way to a pellet. And then we resuspended in an equivalent volume as the BAL fluid, and then add the RNases. So it should be balanced for basically the volume. And the reality is, I didn't show you this data just for time, is that actually there's endogenous RNases in BAL fluid. So if you take a naked RNA, like a naked microRNA, and you shoot it into BAL fluid, it disappears within 15 minutes. So even in the native fluid, there are plenty of RNases there to degrade truly naked or free RNA.
Yeah, but you know that the major diatriba in the field is among vesicle contained RNA and protein bound RNA. So if you just use the RNA, naked RNA, that's not going to
I totally agree with that. So doing the RNase only proves that it's protected, and that could be in a protein complex and it could be in a vesicle. So it's hard because it's very hard to to convince yourself based on negative data. So I've tried to do argonaut immunoprecipitations, which have been shown in circulation to bring down microRNAs. I can precipitate it from circulation. I cannot from BAL fluid. So that's some data to suggest that maybe more of the RNA is in a vesicle fraction rather than a protein bound fraction, but you're right. We haven't shown that for sure there's nothing there. And maybe the way to get at that right is to do a proteinase K digestion first and try to see if we could release RNAs. I think that's really interesting. Actually, somebody in the lab is sort of working on that as well right now.
That could be a way. The other question is, so I really love the human data and the sponge model, but I was wondering, so you said that the clinicians put the sponge in the nose and then they just take it out and you try to flush vesicles out, but what if there are also cells?
Yes, so in this model, so I haven't tested it myself, but our collaborator tells us she doesn't really get cells out of the sponge. I don't know if she's ever tried to section through the sponge to see if cells get trapped in the sponge. But the sponges are basically frozen, and then they will be thawed and harvested. So I don't know that there's a lot of time for a cell that was trapped to sort of secrete vesicles after the fact, but it certainly is an important consideration if that's maybe what you were thinking about.
I'm thinking if the vesicles extracted from the sponge are contaminated with cells, and you know, as you can imagine, even if I'm sure there are not many cells in that biofluid, but even if there were a few, I mean, then you don't know if there is this signal in the vesicles, basically.
Yeah, I mean, I think we haven't done anything to formally exclude that, and we should definitely probably take a second look and see and make sure we don't see a lot of cells. That's basically just sort of what our collaborator has communicated to us. I mean, I don't know. I think it's a really kind of interesting question. I'd be interested to hear what other folks think, because, you know, I feel like having worked in biofluids versus working in cell culture, it's actually kind of hard to get a lot of vesicles out of cell culture.
Yeah.
And so the high concentration that we get in biofluids, man, if the cells are producing that like immediately, I wish we knew how they were doing that, because it might make our cultures a lot better, you know, to actually get EV production. But that's a good point. We should definitely look at it.
Okay, I do have other questions on the tracking EV systems that you showed, but I think I can take that offline with you, because I'm
Sure.
really interested in that.
Sure.
Very nice talk.
Thanks.
So we have a question from Alissa Salim.
Hi, Heather. That was a really nice talk. And my question was about the origin of EVs within BALF. You looked at, and using your mouse model, you looked at epithelial versus immune cells. And unless I missed it, initially you were saying there was no difference between the IMM challenge mice versus the allergic asthma mice. But when you used the mouse model, there was an increase in the EVs coming from the, I think, the immune cells, but not the other ones. So how does total cell number equal each other when there seems to be a specific difference there?
Yeah, I think that's a great question. My feeling or interpretation of this is, because we're seeing increases in what is a small subpopulation, is that our particle tracking is just not sensitive enough to really detect the changes. I think in our hands, we tend to feel like detecting less than a twofold change by particle tracking is kind of challenging, and that would have to be the whole population. So I think that that's sort of how we've thought about it, but it is a good point. And, you know, I will be the first to admit that in our tracking system, I don't think we're seeing anywhere near all of the EVs, not even close, because we're limited basically by detection through the flow cytometer. So it depends, you know, on how much fluorescence is incorporated into each of those EVs. So I think we're getting the tip of the iceberg, but not the whole story. So it'll be great over the next years as both, you know, reporter systems get better, and also, you know, the technology gets better, so that we can actually detect things that have fewer and fewer tags, which, you know, may be more kind of physiologically common, right? We think that any EV can't carry that much of any protein because it's just not that big.
Right. I think you can say that for the whole EV field. It's the tip of the iceberg.
Absolutely.
But yeah, and just a follow. So is there any other cell type that's likely to be found releasing EVs in BALF other than immune cells and epithelial cells?
I mean, the other cell types that are present, and we certainly have not tested them, there are certainly fibroblasts that sort of are in the stromal space. And, you know, in asthma, there's a lot of activity of those cells. So it certainly could be that they're secreting. Whether that's actually making it all the way out into the BAL fluid, I'm not sure, because it's more of a tissue population. And then I know a lot of people have been really interested, right, in endothelial cell EVs. Again, I think it's a great, super interesting question, probably in all types of lung inflammation, especially, you know, in alveolar spaces. You think about it, the apposition of endothelial and epithelial cells is incredibly close, right? Because you really want good gas exchange. So I think those are the other two populations, if I had to make a guess, that might be producing. But really, like, you know, epithelial cells, number one, because there's just so many of them and they're kind of sitting right in that airway lining fluid. And then the immune cells, because they come into the tissue and then they will actually go all the way out into that fluid space as well.
Awesome. Thank you very much again. Great talk.
Thanks.
And we had a question from Carolina.
Yeah, thank you. It's very wonderful talk, Heather. It's just, I think I'm sort of missing the information on the plasma EVs. I'm just wondering whether they are informative for the case of asthma and in comparison with the, you know, the valve, and also if they are sort of differences in asthma patients in the plasma, would they actually have a function and, you know, go to other organs or something like that? Have you looked into that?
Yeah, so great question. Obviously, there's a lot of interest in blood-based diagnostics in asthma, and all I can tell you is for sure the microRNA profile changes dramatically. But again, we use a model where we do an immunization first. So we induce a systemic inflammatory response. And when you induce a systemic inflammatory response, no question, there's enormous changes in the microRNA composition in the blood. So to be able to disentangle that, really the lung-specific inflammation and its effect on circulation, we need a different model, which we could definitely do, where we really begin this immune response in the lung. We don't start it sort of as a systemic response. But I think it'd be very interesting to know. You know, I was really hopeful with our tracking system that we might be able to see epithelial EVs escape out into circulation, and that actually might be kind of a marker of this inflammation. So far, I cannot see that. So again, it's a negative data. It's hard to be sure that that is true, that there never are, you know, epithelial EVs and allergic inflammation that make it out into circulation. But I can't see an obvious population. But that was one of the first experiments that I did, because I thought it would be really cool, right? If all of a sudden, when you had this inflammatory response, that epithelial cells were not only secreting out sort of to the environmentally facing world, but also back into circulation. Yeah. I couldn't see it, but maybe in a different situation or with a different system.
Yeah, maybe sort of like a detection limit also. Absolutely possible.
Plasma is also just so hard. Honestly, the respiratory fluids are way easier to work with than anything. Way harder.
Heather, your diagram, am I on now?
Yeah, you're on.
Yeah, you're on.
Your diagram and other left out outer membrane vesicles, which are there with lots of goodies to, it's very unexplored by our world, our allergic inflammation world. Mm-hmm. And they're certainly in those fluids. And a little bit of LPS goes a long way.
That is absolutely true.
So it will be great. You were Mark Ansel's lab.
That's correct.
So you have fantastic training. He's really a bear.
Yeah, it was wonderful.
But you read a
so
great,
we got to that.
Place now.
Oh.
I'm done.
Sorry, Phil. Sorry. I just wanted to say, I know we're running a little over time. We came to the end of our questions, but I did have one more comment, which was for the discussion you were having with Dolores about whether the RNA is non-vesicular or contained in extracellular vesicles. We've used Triton X100 combined with RNAs to look at that, and that works pretty well. So we could send you our protocol offline if you're interested in that.
Yeah, we've also done that too, because this is an expert group, and our results are consistent with the idea that it's in vesicles. The only problem that I had a little bit was I felt a little concerned that our ability to precipitate the RNA got worse when we added Triton. And so it was—
That's interesting.
I don't know if it was just— that was early days when I had just started my lab. So, you know, things never work as well in the beginning. So I think we should actually go back and redo it. And yeah, we would welcome certainly your protocol to to test. I guess maybe if nobody else has had that problem, it was probably a first 6 months of the lab kind of issue, but I got a little bit concerned about whether we were misinterpreting our result for that. But
Gotcha. I'm not aware of that problem, but I certainly am going to ask my postdoc now.
In addition to outside, free or vesicle associated, there's the problem of on the vesicles and in the vesicles to deal with, where they could be RNase resistant, but on the vesicles.
As possible.
All righty. Well, this was great.
Yes, it was.
Thank you, Heather and Carolina. Thanks for asking me to host. This was a pleasure. Thanks, everybody.